Great, thank you. Welcome back to the BPI Theater. We're going to continue with our programming. So our next presentation is by Dr. Greg Adams from Fujifilm Diosynth. He's the Director of Analytical and Formulation Development at Fujifilm, and his presentation, The Portrait of a Protein, Exposing the Character with Advanced Analytics. Greg.
Opportunity to speak today. So the next 20 minutes, just take a little bit of time and talk about some of the challenges and solutions in characterizing today's fairly complex biopharmaceutical products. But to begin with, just a little introduction if you're unaware of Fujifilm Diosynth Biotechnologies. We're a global CDMO with sites in the UK and 2 sites in Texas and RTP. And we've worked on about 280 different molecules. So I'm kind of drawing on that experience today with the analytical characterization of a multitude of molecules that we work on. So to kind of go over the presentation, talk a little bit about the nature of the analytical challenges and how protein characterization is really being driven by a new era of analytics and regulations, and then the real need for advanced analytics. So the nature of our analytical challenges, proteins are really complicated molecules. They have 3-dimensional structure. You've got to worry about your primary, your secondary, your tertiary structure. You have to worry about them self-aggregating and self-forming covalent molecules and complexes. So these proteins are rarely homogeneous. And so it requires many tests to look into the protein heterogeneity. And to kind of delve into that a little bit, I'm going to talk a little bit about monoclonal antibodies. So again, monoclonal antibodies, they have 3-dimensional structure, they're multiple chains, they're linked by disulfide bonds, they have glycosylation on them, they're divided into multiple subclasses, multiple CDR regions. And so there's a reasonable amount of complexity in something that we think is a fairly simple protein, but it's quite complex. And it requires a whole host of analytics to look at the function of a single protein. And so we need multiple tests to assess the mAb heterogeneity. And so we've got a picture of the mAb here with various different motifs and modifications on there. So on the termini, we have pyroglutamate formation, we have deamidation, we have oxidation, we have truncation. So we need an assay to look at those. And so we use something like charge heterogeneity by imaging IEF, where you're going to start separating out these species that have changed based on charge. Then you get to glycosylation. There's multiple glycans on an antibody, so then we need an assay to do that. So we look at using something like UPLC with Hillick and labeling of the glycans, and you get this profile to look at the glycans. After that, you've got to worry about the molecule clipping, forming half molecules, disulfide shuffling. So again, there's another assay there. So we monitor that by traditionally capillary electrophoresis, SDS. And then after that, you've got all those motifs, and then the molecule starts to aggregate. So then you've got another assay you've got to pile on top of that. So then we use size exclusion UPLC to start looking at the aggregates. So you can see here we've got 4 or 5 different assays just to look at the various heterogeneity. So our analytical philosophy is really driven by wanting an understanding and the expectations of a product's quality attributes. So on those previous slides, there's 5 or 6 quality attributes. Most antibodies, you've got to look at all of them. Some you may not need to. So we want to develop methods that have a line of sight from early process development through all stages of GMP manufacturing. What we really aim to do is have consistent analytical data through the clinical development life cycle. So in many respects, we're really entering a new era of analytics. It's driven by science, it's driven by innovation, it's driven by regulatory factors, new instrumentation, QBD, PAT, biosimilars, and needing to know the differences between an originator and an innovator molecule. And what it's really doing is driving that quality needs to be built into the product. So we need to understand what the analytics are telling us and then manage the expectations of what the molecule is. And then it's going to be driven by business factors. There's pressure to reduce development costs, resources, and timelines. And then for ourselves as a CDMO, we want to be able to differentiate ourselves in the marketplace with the analytical offering. And all of that, again, is feeding back into the focus on critical quality attributes. And so we've got 3 examples here, kind of delving into a little bit more detail from the antibody to things we would see in a E. coli protein to a mammalian cell-expressed protein. There are many motifs, many modifications that these molecules may have. And then we need to meet new expectations for regulations. Last year, an FDA guidance came out that build upon an original guidance on method validation that was published in 2000, and it kind of went a little bit step forward to saying we need to look at the robustness of the life cycle of the molecule. We need to understand the orthogonal nature of those assays. So that means that our approaches to analytical characterization must evolve. We need to go from a one-dimensional picture of our molecule to really the three-dimensional image of the protein. So what that means is that while we may be looking at things like size exclusion chromatography, it's not a matter that we're separating aggregates. Do we know what the size of those aggregates are? If we're doing a peptide map, do we know what the peaks are saying? Do we know that there's sequence variants underneath there? If we're making a glycoprotein, we might get a nice separation of the glycan structures. Do we know whether there's any immunogenic glycans there? So we need to get we need to get under the peaks. We need to have orthogonal analysis. We need to look at it in a three-dimensional manner. So for the rest of the presentation, I want to give you 3 or 4 examples of kind of that next generation analytics, the delving under the original chromatography. Chromatography. So I'm going to touch on 4 different areas of what we'd consider fairly high-end analytics and using them routinely, using them almost on a day-to-day basis. So one is mass spectrometry, and the examples are, is the protein's amino acid sequence correct? Are there sequence variants there? Size exclusion chromatography with multi-angle light scattering. Is the molecule size what we think it is? We just get a peak and we resolve some aggregates. Are they monomers, are they dimers, or is there something unique going on under there? I'm going to talk a little bit about isothermal chemical denaturation. It's a new technique that's really giving us some true insight into how a molecule is folding, and it's really giving us some insight that some of the other biophysical techniques have never told us about whether a molecule is going to be stable or not. And then I'm going to talk a little bit about glycosylation profiling, because we know that if we have the wrong glycan profile, it can impact the yield, it can impact the immunogenicity, it can impact the efficacy of the molecule. So sequence variant analysis has become a very important topic, particularly in the biosimilar arena, but it's really just through clone selection of an originator molecule now. When we create our new molecules and we code the DNA, is it giving us the right amino acid sequence? And then there's multiple examples of various regulatory bodies saying you need to pay attention. This one goes back to 2009 through EMA. So one of the things we want to look at is characterizing sequence variants during clone screening. So what we're looking here is the conformation and characterization of low-level variants that may be present in the protein that's being expressed. 5 years ago, we just did this through SNP analysis, just looking at the DNA. But now we have the techniques to get it to the peptide level and the protein level. But the identification of this is really time-consuming. It's a laborious process, but we have new tools that are coming out through our instrument vendors in automating with higher-end computing to be able to take what may have taken a week's worth of analysis to do it manually, to do it in 25 minutes by computer-assisted algorithms. So I'm going to show you some of that data. And what we're aiming to do is actually get down to below 1% sequence variant analysis. So there's a couple of approaches to this. There's the obvious example where we're doing a mass spec analysis of our peptide map, and you're seeing 2 clear peaks here, and we can clearly see from the analysis that we have a post-translational amino acid substitution. In this case, it was our known variant. So we can key in on the masses of those peptides, and we can create these extracted ion chromatograms, and we can quantitate known levels. And you can see over here, we're talking about levels of 5 to 10%. That's kind of easy to see. The challenge now is what happens when it's below 1%. So then we need to get into an automated LCMS analysis. So you can look at, here's a fairly complicated chromatogram of a monoclonal antibody. There's about 50 different peptides being produced of all different sequences. And so what we're able to do now is use some automated software to process that data. It's going to go through and it's going to globally look at every amino acid in that 150 kilodalton protein, every peptide. Is there an amino acid substitution? And then it comes back with a ranking system based on the MSMS data on how confident we are in that example. So I'm going to show you some of that data. And what we're able to do now is use some automated software to process that data. It's going to go through and it's going to globally look at every amino acid in that 150 kilodalton protein, every peptide. Is there an amino acid substitution? And then it comes back with a ranking system based on the MSMS data on how confident we are in that example. So here's an example that the data shows us. So what you're looking at here is, is a fragmentation of a triptych peptide by mass spectrometry, and each of the little red lines represents an amino acid change. And you can see here there's a change. There's one sequence here of an amino acid. And what we see here, I don't know what's hard to see on the slides. This peptide here from there to there changed by 1 Dalton. And that's a change of a glutamine to a lysine, or an asparagine to aspartic acid. And so the power of the mass spectrometer, it can tell that difference with very high confidence, even if it's just a 1 Dalton change. And this example, in some cases, was as less than 1%. And so you can see here in this little box down here where we're looking at multiple clones, and so now we're looking to see which of these clones have the least amount of that variant, because that's the clone that we want to move forward to for expressing the molecule of interest. So the next example is characterizing SEC species by moles. Moles has become a fairly standard technique. It's going to measure the absolute molecular weight, the aggregation state in solution. So here's an example of a size exclusion chromatogram. We have our main peak, we have a smaller peak here, and then we have another little smaller peak out here. So that's the UV trace, but then we send that through the moles detector, and the moles is going to start telling us what the molecular weight is. So the main peak is 147 kilodaltons. That corresponds with the observed molecular weight by mass spectrometry. So we know that peak is a dimer. We get out here, the next peak is twice that molecular weight, 314 kilodaltons. Then we get another series of size distribution up here at 759 kilodaltons. So it's really clear that we're going from a monomer to a dimer to a pentameric state, which is pretty typical for antibodies. You don't often see it go from a monomer to a dimer to a trimer to a tetramer. The antibodies tend, once they really aggregate, get up into the pentamer versions. And so this is a really key technique to let us know how big those dimers are going to be, in part from the purification of the molecule as well. If we know that we're going to have very large molecular weight species, it might guide what kind of chromatography we use to whether we want to resolve that or whether we want to exclude it. So the next technique I want to touch on is chemical denaturation. This is a new technique that's only been around for about 18 months, and it's looking at the protein stability through chemical denaturation. You're taking a protein, you're putting guanidine into it, and then you're heating the protein, and you're looking to see the unfolded state of the protein in the presence of guanidine. And this is some work that we've been doing with Unchained Labs, who have now commercialized this instrumentation. So it's kind of a little complicated, and I'm just going to give you a little bit of a snapshot of what's happening when we're unfolding a protein. Proteins go through 3 stages of unfolding. They go from their native state as they've been expressed. Then as you denature it, it goes through an intermediate stage where it starts to fall apart, and then you get to the unfolded range. And this is kind of some data that Unchained Labs kind of put out, showing that the various forms of how this denaturation occurs. And then by monitoring it with chemical denaturation, you can get these plots, and it's called DeltaG Trending. You're looking at the change in enthalpy as the molecule is unfolded. And as the molecule unfolds in terms of protein concentration, you can predict the stability of the molecule. So I'm going to kind of show you, this is kind of showing you, the data output. So what we're looking at here is a concentration of guanidine, and then the fluorescent signal of the protein as it is unfolding. And you see it going through these 2 different transition states here. There's one state here, and there's another state here. So this is showing you going from the native state to the intermediate state, then the intermediate state to the unfolded state. So what we now start to do is to start to use this technique for buffer screening. And so here's a fairly complicated example, but what I want to show you here is looking at protein concentration of an antibody in 2 different buffer types. And what we have across the bottom here is protein concentration, and you're looking at what is happening as the protein is unfolding. Now, if you'd have just done this at 1 concentration, you would have chosen buffer 2 as your most stable protein because it had the higher delta G. It's showing that in its native state that it's going to be unfolding. And what happens when we look at the difference in protein concentration is this gap starts to become bigger based on its concentration dependence. So if it had done this study at just one form, at one at one protein concentration, we would have concluded that buffer 2 was the most stable buffer. But as you go higher in protein concentration, it's showing that that buffer has a higher propensity to aggregate. So in this case, we would go back and use buffer 1, and that bore itself out in later accelerated stability. This data we don't see through any of our other biophysical characterization techniques. So in the last couple of minutes, just to talk now a little bit about Glycan characterization and some of the newer aspects of Glycan identification and then automated data analysis. So we're using some tools, but using mass spectrometry and UPLC, where we're taking our Glycan profile here, and then by getting mass spectrometry data, and then on this side of the slide is a dextran ladder, and there's a database now that's put out by an organization called Nyberg that has correlated to this dextrose ladder what all the main Glycan GU values would have. And then we can take that to the next level and then go into mass spectrometry and do identify what those glycans are. So this is all an automated data analysis where we're doing an LCMS analysis of the released glycans. It's about a 25-minute chromatography run and then about an hour of data processing by mass spectrometry to get this report that's identifying the various glycans based on its GU index and based on the mass. So now we can start to look at, if it's sialylated, do we have any immunogenic sialic acid there? Do we have glucnac or glunac? And then the data output is very automated and it's very accurate as well. Without this data analysis would be taking a week of analysis, working through the mass spectrometry, getting the right masses. Now we have a very automated process. So if we're going through and we're screening 20 or 30 different clones, we can do this in a day rather than a week. And so here's 3 examples of the Glycan identification. This is, again, is all automated. This is the report that it's printing out. So you can see here an example of a mAb and the Glycan species that you would expect from monoclonal antibody. An example of a non-mAb protein with very high manostructures. So you just see the nice manostructures where you're going from man 5, 6, 7, 8, and 9. And then when we want to get really complicated, we can get to very highly sialylated species. So here you can see out at the end here, you're getting to tetraantennary structures, fully sialylated. So these are very, very large molecular weight, very complicated sialylated species, and we're able to automatically identify all these very accurately. So in conclusion, what we really want to do is targeted successful application of all of these analytics. And so here's an example where we've been using glycosylation biomass spectrometry to monitor what's happening at the various scales. And so what you're seeing here is a comparison in a 110-liter stainless steel reactor to a 200-liter single use to a 1,000-liter single use. And you're seeing that the glycan profiles are not changing, and then we're getting consistent growth and consistent titer through our product. And in this example, we ended up scaling this up to even larger than 1,000 liters, and we were able to demonstrate that we can manufacture the same protein at multiple sites within our network and get consistent analytical profiles and consistent product growth and quality, and that's what we're trying to aim for. Thank you. Any questions?